tissues, and the blastocyst starts to embed in the endometrium during day 8. Lacunae appears in the syncytial trophoblast during day 9. Lacunar network formed by fusion of adjacent lacunae during day 9. The blastocyst sinks beneath the endometrial epithelium, and the surface defect is filled by a closing plaque during day 10. I'm jumping. The syncytial trophoblast erodes maternal blood vessels, that is the sinusoids. I told you what is sinusoids. The sinusoids are just like capillaries, but they are wider and engorged with blood, so they are called sinusoids. So syncytial trophoblast erodes maternal blood vessels, allowing maternal blood to seep in and out of lacunar network, thereby establishing a utero-placental circulation during day 13. Primary chorionic villi develop during day 13. Primary chorionic villi during day 13. Precaudal plate develops during day 14. I told you about the precaudal plate. Precaudal plate. Now, at the beginning of now, summary of the second week again. At the beginning of second week, the blastocyst is partially embedded. The trophoblast differentiate, again I'm repeating, into an inner cytotrophoblast and an outer syncytiotrophoblast. Picture, this is the endometrium. These are the sinusoids. This is the connective tissue of the uterus, connective tissue, and this one is the surface lining epithelium. Here you can see simple columnar in nature, and these are the uterine glands. Uterine gland, the whole thing is lined by epithelial cells. And so this is endometrial stroma. Stroma means connective tissue. So this whole embryoblast has sinked into the maternal, into the wall of the uterus. There is a surface defect here. It has closed during day 10, as I told you. And this is our embryo. These tall blue colored cells are epiblast, and the yellow colored cells are hypoblast. And as this is the second week, we call it a germ disc, and we call it a bilaminar germ disc. What does it mean? It means it has two layers, the epiblast and the hypoblast, so bilaminar. And the outer layer, the trophoblast, this was the inner layer. If you remember, embryoblast or inner cell mass, and this was the outer cell layer. So outer cell layer also divides into two. The inner cells, look at the cells, these are the big cells, and we call it cytotrophoblast, and the outer layer, green in color, where the cell boundaries are not seen. Look here, no cell boundaries are seen, so we call it syncytiotrophoblast. And the whole sheet of cells, this is a sheet of cells because we cannot see the cell boundaries, only the nuclei are seen. We call it syncytiotrophoblast, and the syncytiotrophoblast has the quality of erosion or eroding maternal tissue. That means it erodes inside the wall, and this cavity is the amniotic cavity, and this big cavity is still the blastocystic cavity, if you remember about the blastocystic cavity. So I think this is the picture of second week, that is the starting of the second week, that is day 8. So this is the bilaminar germ disc, epiblast and hypoblast. And these are the maternal blood vessels, the sinusoids. Now, so by 9, lacunae develop in the syncytium. As I told you, lacunae develop in the syncytium. So look, this is day 9 day of 9th day, and look at the lacunae here. If you can see the spaces in the syncytiotropoblast, 1, 2, 3, 4. These are the lacunae which appear during day 9. And look at the bilaminar germ discs, the epiblast, the hypoblast, the blastocystic cavity, the amniotic cavity, and the lacuna or the spaces appear in the syncytium or the syncytiotropoblast during day 9. Subsequently, maternal sinusoids are eroded by the syncytiotropoblast. As I told you, the syncytiotropoblast has the ability of erosion. They can erode maternal tissue, maternal blood vessels, they erode maternal blood vessels, and then what happens? Maternal blood enters the lacunar network. So look here, these red spaces, these are all lacunae, and lacuna, they intercommunicate with one another, and so they form a lacunar network, and the green color tissue, the syncytiotropoblast, they erode maternal blood vessels, and so maternal blood sips into the lacunar network, and you can see the lacunar network, they are all filled with maternal blood and they are red in color. So this is about day 9. Now by the end of second week, that was important, that's why I told you again, by the end of second week, that is 13th day, a primitive uteroplacental circulation begins. What does it mean? It means this circulation, the blood here, and blood coming from the maternal sinusoids into the lacunar spaces, the placenta will form here. So this is, we can say that during 13th day or day 13, a primitive uteroplacental circulation has established during day 13, maternal sinusoids. Meanwhile, the cytotrophoblast forms cellular columns penetrating into and surrounded by syncytotrophoblast. These cellular columns are primary chorionic villi. So let us see the figure. What happens? The cytotrophoblast here, look at the cytotrophoblast, these cells, the cells here, less green in color. So they form cellular column, like here, 1 column, 2 column, 3 column. And the cellular column, finger-like projection, they are surrounded by syncytiotrophoblasts. So we call it a primary villi. So during day 13, the cytotrophoblasts proliferate and they form cellular column, 1, 2, 3, 4, in the near to the embryonic pole. This one is the embryonic pole, and this one is the embryonic pole I told you. And so, and this cytotrophoblastic column of cells, they are surrounded by syncytiotrophoblasts, and now we call it a primary villi. So the cytotrophoblasts form cellular columns penetrating into and surrounded by syncytiotrophoblasts. These columns of cytotrophoblasts are nothing but the primary chorionic villi. So you can see the primary chorionic villi or primary villi simply. By day 8, the inner cell mass or embryoblast differentiate into, I'm repeating, epiblast and hypoblast. Epiblast and hypoblast. So this is another figure taken from another book. It is not in a can buy. So this is taken from another book. So this is epiblast, this is hypoblast. So a bilaminar germ disc. This one is the amniotic cavity, this inside trophoblast, and this is the cytotrophoblast, and this is the uh, blastocystic cavity. Epiblast gives rise to amnioblast. Line the amniotic cavity is superior to the epiblast layer, as you have seen. Endoderm cells are continuous to the exocoelomic membrane, and together they surround the primitive yolk sac. I told you the blastocystic cavity, when surrounded by exocoelomic membrane, a membrane, this is exocoelomic membrane, a membrane secreted by the hypoblast. I'm telling you again, this is the blastocystic cavity. This is hypoblast. Hypoblast secretes a material which form a membrane, which is called exocoelomic membrane. Exocoelomic membrane. Now this cavity, the blastocystic cavity surrounded by this exocoelomic membrane is known as the yolk sac. So the blastocystic cavity is converted into yolk sac when the hypoblast secrete the exocoelomic membrane. By the end of second week, extraembryonic mesoderm, I told you, fills the space between the trophoblast externally and the amnion and the exocoelomic membrane internally. I'm showing you a figure. So this is our extraembryonic mesoderm. So extraembryonic mesoderm covers the cytotrophoblast externally and the amniotic cavity and the yolk sac cavity internally. So this one is the exocoelomic cavity. This one is the, when vacuoles appear in the extraembryonic mesoderm, the extraembryonic coelom or chorionic cavity is formed. So this extraembryonic mesoderm, within the extraembryonic mesoderm, vacuoles appear here. You can see this is taken from Langman. I'm showing you the figure again. So look at this, the end of second week, the whole embryoblast has embedded fully into the wall of the uterus. And this is the lining epithelium of the uterus, and the surface defect of the wall has been closed by blood clot, and then there will be regeneration of surface epithelium. Look at the primitive yolk sac. I told you blastocystic cavity, when covered by exocoelomic membrane secreted by the hypoblast, that is, hypoblast secretes a membrane-like material which covers the entire blastocystic cavity. We call it an exocoelomic cavity. And this and here is the epiblast amniotic cavity. Now, I was talking about the extra embryonic mesoderm. What is that? These cells, loose cells, the white color here, they fill up the cytotrophoblast externally and the exocoelomic cavity and this the epiblast and hypoblast here, and we call it the extra embryonic mesoderm. And ultimately what happens, vacuoles appear here. Vacuoles means cavities. Suppose here, 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 and the vacuoles fuse. The vacuoles coalesce. They form a huge cavity. We call it extra embryonic cavity, or we call it chorionic cavity. So here will be the extra embryonic cavity, extra embryonic cavity, or the extra embryonic coelom. Coelom means cavity. Look here, look at the picture. Here, the extra embryonic coelom or extra embryonic cavity. Now there is a question. So we have finished the second week because I talked about the second week previously. Today it was, I just summarized, tried to summarize, I jumped, I understand. So just here, I just repeated because of the I want you to be hammered so that you can remember and you read and study. Now there is a question very common which comes in the prof exam in SAQ. Why the second week of development is known as the week of twos? You can write it and you write in your copy and you have to submit it. I will tell Dr. Masoom to collect it from you. So you just write it. Why the second week of development is known as week of twos? I'm giving you here the answer. So second week of development is known as the week of twos because the trophoblast differentiates into two layers. Trophoblast differentiates into two layers. Number one, the cytotrophoblast internally and the syncytiotrophoblast externally. Trophoblast is the outer cell layer, if you remember, and embryoblast was the inner cell layer. Now, during second week, the trophoblast differentiates into two layers, the cytotrophoblast and the syncytiotrophoblast. The embryoblast, that is the inner cell mass from which the embryo develops. The embryoblast also differentiates into two layers. What are those? The dorsally, the big cell, tall cells, epiblast, and ventrally, the hypoblast. EP means above, hypo means below. So epiblast and hypoblast. Two cavities are formed, the amniotic cavity dorsally and the yolk sac cavity ventrally. Remember, two cavities are formed, the amniotic cavity and the yolk sac cavity. It was blastocystic cavity, then it was the exocoelomic cavity or primary yolk sac, and then there is the secondary yolk sac or the definitive yolk sac. Just remember yolk sac cavity. Primitive yolk sac or cavity or exocoelomic cavity. When the I'm just explaining about this primitive yolk sac, exocoelomic cavity. I'm explaining this. So primitive yolk sac or cavity, or which is also called the exocoelomic cavity. This is nothing but the blastocystic cavity. If you remember about the blastocystic cavity, when it is surrounded by exocoelomic membrane, we call it primitive yolk sac or primitive yolk sac cavity or exocoelomic cavity. So blastocystic cavity, when lined by exocoelomic membrane, a membrane secreted by the hypoblast, we call it a primitive yolk sac cavity or exocoelomic cavity. Look here. So this was basically the blastocystic cavity. This is hypoblast, yellow cells, they secrete the exocoelomic membrane. When the blastocystic cavity is covered by exocoelomic membrane, we call it exocoelomic sac or cavity, or one has two names, or the primitive yolk sac. And what happens here, this and the hypoblast, then ultimately it will secrete endodermal cells and they will form another cavity here like this, lined by endoderm. I'm telling you again, look at this figure. Hypoblast secretes exocoelomic membrane or Husser's membrane, the name of the scientist who discovered about this membrane. So this is also known as Husser's membrane. we call it exocoelomic cavity or primitive yolk sac. Now, this primitive yolk sac, these hypoplastic cells will form endodermal cells, endoderm, and they will create one cavity like this here, like this. So now we will call it a secondary yolk sac. And the other cavity here that will ultimately pinch off from here and will form a cyst. We call it exocoelomic cyst. Forget about this. Just remember blastocystic cavity lined by exocoelomic membrane form the primitive yolk sac or yolk sac cavity. Then the hypoblastic cells, the cells from hypoblast come and line another cavity here, which is the endodermal cells. We call it yolk sac cavity, secondary yolk sac cavity. This is important. The secondary yolk sac is important because later in life we will see our gut tube will be formed from this secondary yolk sac, our gut tube. Our gut tube, the alimentary canal or the GI tract, whatever you say, formed from this secondary yolk sac lined by endoderm here. So, আবার এটা আসছে. Yeah, 4 people are waiting. Now, secondary yolk sac or definitive yolk sac, the other name is definitive yolk sac when the primary yolk sac is lined by endoderm. Just remember, what is a secondary yolk sac? The other name of the secondary yolk sac is definitive yolk sac. What is that? When the primary yolk sac is lined by endoderm. So in other way, you can say when the primary yolk sac is lined by endoderm, we call it a secondary yolk sac or a definitive yolk sac. This is very important because I told you the gut tube will be formed from here. Now I'm going back to the week of twos. Why it is called week of twos? So the extraembryonic mesoderm splits into 2 layers. What are these? Somatopleuric or somatic mesoderm and splanchnopluric or splanchnic mesoderm. The extraembryonic mesoderm, I'm giving emphasis, extraembryonic mesoderm. It's not embryonic mesoderm. This is extraembryonic mesoderm. This is divided into 2 layers, the somatopleuric or somatic layer and the splanchnopluric or splanchnic layer. So the somatopleuric or somatic and the splanchnopluric mesoderm. Oh, important night in the session. So this question, why the second week of development is known as the week of twos. I'm giving you an assignment. Why the second week of development is known as the week of twos. You just write it down and submit it. I will talk with Dr. Masoom. We have finished the second week. Now I will switch on to the third week of development. The third week of development is known as the trilaminar germ disc. So the second week, 2 for 2. Second week, the germ disc was bilaminar, 2 for 2. Here, the third week, that is week 3, 3 for 3. The germ disc is trilaminar. Remember in this way. So the trilaminar germ disc. So what are the events of third week? Number one, you try to remember the events, first, second week, third week, write it down. So number one event of third week starts from day 15, right? So 15 to 21 day. So number one is the formation of primitive streak. Formation of primitive streak. Number two, formation of 3 germ layers Formation of 3 germ layers. This is important. And we will ask you, what are the 3 basic germ layers? The germ layers. So the process of formation of 3 germ layers is known as gastrulation. The process of formation of 3 germ layers is known as gastrulation. And this is the most characteristic event of 3rd week. So it becomes trilaminar. That is why I told the 3rd week of development is also known as the trilaminar germ disc developmental layer week. So formation of 3 germ layers. Number 2, 3, formation of notochord from the notochordal process. This is also important for human. We have a vertebral column, and notochord is like a rod and like an axis around which the axial skeleton develops. So just remember the events. 3rd event is the formation of notochord. From where? From the notochordal process. Number 4. Number 4 events of the 3rd week is formation of cardiogenic area. Formation of cardiogenic area. Remember, in the cardiogenic area, our heart will develop. Number 5th event, formation of allantois. Just remember it, because humans, we don't have significant effect of this allantois. Just remember. Number 6, formation of neural tube. This is important. And neural crest. And the process by which neural tube is formed is known as neurulation. In my first lecture class, I remember I talked about neurulation, gastrulation, if you remember. So number 6 event is the formation of neural tube and neural crest, and we call it, the process is known as neurulation. Number 7, this is important, formation of somites. Somites formation occurred during day 20. Day 20, that is the end of 3rd week. Day 20, that is the end of almost the end of 2nd week. Somite is very important. Now, number 8 event, formation of intra-embryonic coelom or intra-embryonic body cavity. So during 2nd week, there was the formation of extra-embryonic coelom or extra-embryonic body cavity. And during day 3rd week, there is the formation of intra-embryonic coelom or intra-embryonic body cavity, or some of the books, in some of the books it is written secondary body cavity or definitive body cavity. So intra-embryonic coelom or intra-embryonic body cavity is also known as the secondary body cavity. Number 9th event is the formation of somatic and splanchnic mesoderm from lateral plate mesoderm. So in the 2nd week, there was formation of somatic and splanchnic mesoderm. That was extra embryonic mesoderm. And here in the 3rd week, there is the formation of somatic and splanchnic mesoderm from the lateral plate mesoderm, which is a part of intra-embryonic mesoderm. There is difference, intra-embryonic mesoderm, and that was extra-embryonic mesoderm, remember. Then what happens during 3rd week? What is the 10th event? There is vasculogenesis and angiogenesis. Vasculogenesis means blood and blood vessels form de novo, and angiogenesis means blood vessels sprouting from pre-existing blood vessel. So there is vasculogenesis and angiogenesis. That means Blood vessels and blood forms de novo from mesenchyme, and there is angiogenesis. Angiogenesis means there is sprouting of blood vessels from pre-existing blood vessels. Okay. Now, 11th events, completion of chorionic villi formation. If you remember, I told you during day 13, there is the formation of primary villi. What was it? It was column of cytotrophoblast surrounded by syncyte eutrophoblast. If you remember up to this. Now, during third week, there is the formation of secondary villi during day 16, and there is the formation of tertiary villi during day 20. Remember it. So the villus formation ends during third week. During day 13, there is the formation of primary villi. Day 16, secondary villi are formed. Day 20, tertiary villi are formed. So these are the events of third week. Now, the most characteristic event occurring during the third week is gastrulation. I told you the most characteristic event is gastrulation. Now, what is it? I already told you gastrulation, it is the process by which all the three germ layers are formed or established. So this comes in exam. Write short notes on gastrulation, or maybe what is gastrulation. So in the Viva also we ask you what is gastrulation. So what is gastrulation? It is the process by which all the three germ layers within bracket you have to write ectoderm, mesoderm, and endoderm. So these are the three basic germ layers from where a human develops: the ectoderm, mesoderm, and the endoderm. So gastrulation, we know about gastrulation. Or you can say it is the formative process by which three germ layers are produced, all the same. During gastrulation, the bilaminar embryonic disc is converted into a trilaminar embryonic disc. So embryonic disc was bilaminar. During second week, it was epiblast and hypoblast. And during third week, it will be converted into trilaminar embryonic discs. What are these layers? From dorsal to ventral, ectoderm, mesoderm, and endoderm. From dorsal to ventral, ectoderm, mesoderm, and endoderm. So how gastrulation starts or begins? Gastrulation begins with the formation of the primitive streak. I told you while I was talking about the events of third week, the number one event I told you, formation of primitive streak, and gastrulation is the second event. So how this gastrulation occur? Gastrulation of primitive streak, what is it? So what is on the dorsal and caudal aspect of the bilaminar embryonic disc? So primitive streak, it is a narrow groove where on the dorsal aspect embryonic disc. Let us see. So forget about this one. Just look at this. This is a bilaminar embryonic disc. It's not round. It's the shape is like an egg, but the layers are two. So it is bilaminar, and above is the amniotic cavity, and below is the yolk sac cavity. So look here. This is the primitive streak. It is a groove that appear on the dorsal, because this is the dorsal aspect, and dorsal and caudal aspect of the bilaminar embryonic disc. So what is the primitive streak? It is a groove which appear on the dorsal and caudal aspect of the embryonic disc. Now, this is another thing. I'm talking about gastrulation. So in talking about gastrulation, there are some terms, that is the primitive streak, and so many things will come. So gastrulation, here what happens, the epiblast through the process of gastrulation is the source of all the three germ layers. Remember it. The epiblast and hypoblast are the two basic layers of the germ disc during the second week. Now, I told you that during gastrulation in third week, the three germ layers, basic layers, ectoderm, endoderm, mesoderm, and endoderms are formed. So how they are formed? Number one, for them to be formed, there should be the appearance of a primitive streak. What is that? I told you. Now, I'm telling you another thing. From where the three germ layers will come? The three germ layers will come from the epiblast. So the epiblast, to the process of gastrulation, because gastrulation is a process, it takes time to occur. So to the process of gastrulation is the source of all the three germ layers. That is the ectoderm, mesoderm, and endoderm. The cells of these three germ layers will come from epiblast. And cells in these germ layers, again, I'm writing in the exam, we always ask you, tell me the basic three germ layers. This is very basic question. If you cannot answer, if you have to answer. Like the basic tissues. What are the four basic tissues? Epithelial tissue, connective tissue, muscular tissue, and nervous tissue. This is very basic in histology. And in embryology, the basic germ layer, ectoderm, mesoderm, and endoderm. Okay, so cells in these germ layers will give rise to all the tissues. That is, all the four basic tissues. And when the tissues are formed, all the organs in the embryo, like stomach, like heart, like intestine, everything. So there should be this basic three germ layers. Now, during this period, that is when the gastrulation is happening or occurring, the embryo is called a gastrula. So when gastrulation is occurring, the embryo is known as a gastrula. Now there is a question, what is gastrulation? Write it down, what is gastrulation? So you know, this is a process by which three germ layers are formed. Within bracket, you have to write that. And write short notes on gastrulation. So these are the two basic questions which comes in exam. Write short notes on gastrulation. Now I will talk about the primitive streak now. So this is the first event of third week, the primitive streak. What is this? I told you the first sign of gastrulation is the appearance of primitive streak. I just told you. And this is the embryonic disc here. Look here, the dorsal aspect, we are seeing from the dorsal aspect, that is the epiblast, and underneath is the hypoblast. And here is our primitive streak. It is a linear groove on the dorsal and caudal aspect of the bilaminar embryonic disc. Now, at the beginning of the third week, an opacity formed by thickened linear band of epiblast, thickened linear means straight, band of epiblast, the primitive streak, appears caudally in the median plane of the dorsal aspect of the embryonic disc. So if you remember the figure, you can write that this is a thickened linear band of epiblast which appear in the midline or median plane in the dorsal and caudal aspect of bilaminar embryonic disc. This is a real figure, the third week embryonic disc. Just look at the primitive streak in the median plane figure. As the streak elongates by addition of cells to its caudal end, its cranial end proliferates to form a primitive node. So when the streak elongates, the streak will enlarge, and here there will be the formation of a node, clump of cells known as primitive node. So concurrently, a narrow groove, primitive groove, develops in the primitive streak that is continuous with a small depression in the primitive node, the primitive pit. So I'm telling here. Number one, the primitive streak, a linear thickened band of epiblast. Okay. Now, as cells are added here in the caudal end, it will elongate. This primitive streak, it will elongate, and here there is a clump of epiblast cells. We call it primitive node. So primitive streak and primitive node. Then the primitive streak will go inward. Go inward, we will call it a primitive groove. The primitive streak will go look at the arrow, the dark line. The cells of the epiblast go inward, and we call it a primitive groove. At the same time, in the primitive node, cells will go invaginate. We call it invagination of cells. That is, cells go inward. We call it primitive pit. That is what I was talking about, that as the primitive streak elongates by addition of cells to its caudal end, its cranial end proliferates to form a primitive node. Now, and then a narrow groove, we will call it primitive groove, develops in the primitive streak. That is, this groove, this groove is continuous with a small depression in the primitive node, the primitive pit. Pit means ditch. So look at this figure, this is important. So this is still a bilaminar embryonic disc, but we are in third week. This is the primitive streak, and the primitive streaks, how it is formed, I told you. There is primitive node, in groove develop in the primitive streak by invagination of epiplastic cells. We call it primitive groove, and here there will be primitive pit or primitive, yes. Now the primitive groove and pit result, why they are formed? I told you, they're formed from the invagination of epiblast cells. There are two terms, invagination and evagination. Invagination means inward movement, and evagination means outward movement. So forget about the evagination, as we are talking about the invagination. So what happens? This groove, look here, this is a cross-sectional view of the caudal end of the embryonic disc near to the primitive streak. So this is our primitive streak, and this is our primitive node. What happens? Why this primitive groove appear here? Because the cells of the epiblast, which forms the primitive streak, they invaginate, that is, go inward. Look at the arrow. Cells of the epiblast here, they invaginate. They invaginate, so there is a formation of primitive streak and primitive pit. So they result from invagination of epiblast cells. As soon as the primitive streak appears, it is possible to identify the embryo's neocaudal axis. This is important. Its cranial and caudal ends, its dorsal and ventral surfaces, and its right and left sides. You can understand by seeing this. Slide. Now, when this primitive streak is formed, we can say that this is the bilaminar germ disc. And when this primitive streak appear, we can say that this is the cranial end of the embryo, this is the caudal end of the embryo, this is the dorsal aspect, and underneath is the ventral effect. This is the right part, this is the left part. That is what I was telling, that as soon as this primitive streak appears, it is possible to identify the embryo's craniocaudal axis, its cranial and caudal ends, its dorsal and ventral surfaces, its right and left part. Now there is a question, write short notes on primitive streak. Marks allocated can be 3 or 4. Write short notes on primitive streak. Now, shortly after the primitive streak appears, cells leave its deep surface, I have already told you, and form mesenchyme. You know about the mesenchyme. Mesenchyme, what is it? This is nothing but embryonic connective tissue. Now, shortly after the primitive streak appears, cells leave its deep surface to form mesenchyme. The mesenchyme is a tissue consisting of loosely arranged cells suspended in gelatinous matrix. What is mesenchyme? Mesenchyme is embryonic connective tissue. The cells are loosely arranged here. That is, cells are loose. And in between the cells, there is gelatinous matrix. So these cells which invaginate inward, look at the red sectional view of the caudal aspect, internet connection is unstable. At the caudal aspect of the embryo, and look here, the epiblast cells are invaginating the red cells, and look at the hypoblast, the yellow cells, and these cells are mesenchyme, these red cells, they come from epiblast. They are mesenchyme, and they will form, then some of the mesenchymal cells will become mesoblast. So what is mesenchyme? Primitive streak leave its deep surface and form mesenchyme, a tissue. So you know about this. And look here, the epiblast cells invaginate. There is an interesting thing here. I will tell you in language later on. Just try to understand from here. This is the primitive streak, and the cells are invaginating. Look at the arrow, and there is so there is the formation of primitive groove and primitive pit. Now these red cells, they form mesenchyme. Okay, some cells will become mesoblast, and they will form mesoderm here, ekhane. And some of these epiblast cells will displace these hypoblast cells. Hypoblast means yellow colored cells here. And they will become, when they displace the hypoblast cells, they will become endoderm. And the remaining epiblast cells will form the ectoderm. So epiblast is the source of all the 3 germ layers. What are those? Ectoderm, mesoderm, and endoderm. They are formed from the primitive streak through the process of gastrulation. So look here again, this figure is very important. You have to draw it. What is happening? I'm telling you again, this is primitive streak. Primitive streak is nothing but proliferation of epiblast cells. This is the dorsal aspect, and this is the ventral aspect of the embryonic disc. So cells from the epiblast, they invaginate. Look here, and they form mesenchyme. Some of the mesenchyme becomes mesoblast, and they form mesoderm. Some of the epiblast cells displace the hypoblast cells and become the endoderm. The remaining epiblast cells form the ectoderm. So epiblast is the source of all the 3 germ layers. Now, I will talk about the mesenchyme. What is mesenchyme? This is embryonic connective tissue. So there are some characteristics of mesenchyme, like mesenchymal cells are amoeboid and actively phagocytic. What is amoeboid? They can move through pseudopod like amoebae. So mesenchymal cells are amoeboid and they are phagocytic also. Mesenchyme forms the supporting tissues of the embryo, such as most of the connective tissue of the body of the embryo and the connective tissue framework of glands. So to be a gland, what should be there? At the gland hote gele ki lagbe? There should be epithelial cells, there should be connective tissue cells, stroma, which we call stroma. Now, mesenchyme forms the supporting tissues of the embryo, such as most of the connective tissues of the body and the connective tissue framework of the gland. Mesenchymal cells derived from primitive streak can migrate widely. This is important. They can migrate. They can go from here to there, like the neural crest cells. I will tell you later. Neural crest cells, they will form from neural tube, but they can migrate. The mesenchymal cells derived from the primitive streak, they can migrate. And the mesenchymal cells are the pluripotential cells. If you remember, pluripotential cells means partially differentiated cells, not fully differentiated cells, like erythroblast. From erythroblast, erythrocyte will come. The erythrocytes are the blood cells. They're the fully differentiated cells, but the erythroblast is partially differentiated cells. I told you. These pluripotential cells, mesenchyme, have the potential to proliferate and differentiate into diverse types of cells, such as they can become fibroblast, they can become chondroblast, and they can become osteoblast. So these mesenchymal cells, the connective tissue cells, they can become, they are pluripotential cells. They can differentiate into different types of cells, like they can become fibroblast. What is fibroblast? You know, this is the major connective tissue cells which can form the fibers of the connective tissue. They can become chondroblast, that is the main cell of the cartilage, and they can become the osteoblast, that is the bone forming cells. Now there is a question, write short notes on mesenchyme. So this is the end of this class today. In the next class, I will talk about formation of 3 germ layers. Second event of third week. Second event of third week. In the next class, maybe in the next Tuesday, inshallah, I will talk about the formation of 3 germ layers. Second event of third week. Okay, thank you.
Assalamualaikum, ma'am.
Thank you, ma'am. Assalamualaikum, ma'am.
Assalamualaikum, ma'am.
Assalamualaikum, ma'am.